Today's presentation was related on the phase 1 study that we have on atezolizumab monotherapy in triple-negative breast cancer patients. This study that was in metastatic breast cancer patients had evaluated previously which patients had the best outcome with our immune checkpoint inhibitor atezolizumab. We had seen that patients that had PD-L1 positive tumors were the only ones responding to treatment and also displayed the longest overall survival. But we did further work on the biology of this clinical study to try to understand what would be possible mechanisms of resistance that should be targeted to improve the outcome of this patient population. And what we found is that patients that have activation on the IL-6 and CRP pathway, they tend to have a reduced clinical outcome. Very interestingly, when we perform a preclinical mouse model combining anti-IL-6 receptor with anti-PD-L1, we found that the response rate to this combination increased dramatically compared to single-agent activity in these mouse models. Interestingly, in our company, we have not only the anti-PD-L1 that is atezolizumab, but there is an anti-IL-6 receptor that has been widely used in the inflammatory disease, that is atezolizumab. So the goal would be to move forward in a combination of anti-PD-L1 atezolizumab and the anti-IL-6 receptor tocilizumab to improve the clinical outcome of this patient population. Also importantly is that we don't think that this is a mechanism of resistance specific for the triple-negative breast cancer patients treated with atezolizumab, or not even for immune checkpoint inhibitors as a class action, but rather a phenomenon that happens across tumor types and different type of therapies. So if we are able to combine anti-inflammatory drugs with immune checkpoint inhibitors or other type of therapies, I feel that we are going to give this hard-to-treat patient population a new hope. The toxicity for the single-agent atezolizumab is quite manageable. It's similar to what we have been describing before to other atezolizumab-treated patients. This has been amply described in our publications led by Dr. Licia Emmons in the JAMA Oncology 2019. The activity of checkpoint inhibitors in metastatic triple-negative breast cancer is not as high as the one that we have observed with the standard of care chemotherapy. So the idea is to improve the outcome by leading combinations with cancer immunotherapy. On that note, we as a company in Roche and Tech, we have read out a phase 3 clinical study where we combine nab-paclitaxel with atezolizumab. And this is in fact the first cancer immunotherapy regimen that have improved the clinical outcome of triple-negative breast cancer patients in the last 30 years. So we're very excited that we're moving forward. The next step for cancer immunotherapy in triple-negative breast cancer is combination therapy. In that aspect, we have combined immunotherapy with nab-paclitaxel, that is a chemotherapy, and we have found that the patients that bear PD-L1 positive in their tumors, they have an enhanced clinical benefit both in progression-free survival and overall survival, and this is something that had not been observed in the last 30 years of treating this patient population.